Let's get to it. Okay, so thanks everyone for joining us today. I'll be talking about a highly collaborative project, right? So this is the kind of work that for which you need a lot of people. And in this work, particularly, I've been relying a lot on the work of Julius Fredrickson and the Aphasia lab. We're basically using all their data to do analyses of agrammatic performance. So let's get going. What do we think we know? So agrammatic performance by speakers post-stroke has been noted basically since the mid-19th century formally, right? And the main thing that was noted at first was lack of function words and bound morphemes, so the little linguistic markers. Speech is very reliant on noun production, and there seem to be some verb specific problems as well. Some of those may have to actually do with verb argument structure complexity. So how complex are the representations of particular verbs? And it seems to be the case that speakers who have agrammatic output have particular trouble with these grammatical markers and complex structures. Simplified sentence structure has also been noted, though not always along with this deletion of morphemes. So it hasn't been entirely clear whether this is a separate problem, though probably highly correlated, or whether it's just a necessary part of what might be called a syndrome of agrammatic aphasia. Something else that's been mentioned is what is called paragrammatic performance, and there the discussion is whether we're really talking about something different. So here we're talking about speakers who not so much delete function words and bound morphemes, but they substitute them, so they make a lot of errors. And usually they're also more fluent than the other type of agrammatic speaker. But it's of course possible that these are just two sides of the same coin, that those two output versions are just different kinds of adaptation to the same underlying problem. Another thing that's been noted throughout the history of people writing about agrammatic performance is variable performance. So it's not the case that everybody who has this issue performs in the same way. And even within subjects and between participants in studies, performance has been highly variable, causing some people to doubt the existence of an actual syndrome. Actually, a lot of people doubt the existence of a syndrome of agrammatic performance. Then something else that was highlighted particularly after the rise of, or the re-rise, I should say, of formal linguistics, transformational grammar in the 60s, and in the 70s there's a renewed interest in syntactic comprehension deficits. And there the same discussion is this always the case, that these speakers who have this impaired output always have a syntactic comprehension problem, or is it just a separate issue? We're interested in this also because we want to know about the neural correlates of syntactic processing. So what do we know from functional neuroimaging? There's quite a body of work looking at neural correlates of syntactic production and processing, and basically the findings converge on a network that includes Broca's area, inferior frontal gyrus, and posterior superior temporal cortex, and then sometimes anterior middle temporal lobe is included in that, and there seems to be something going on in premotor cortex as well that might have to do with complex syntactic processing. Lately, there's been a lot of interest in what could be called a dorsal connection between the posterior superior temporal gyrus and inferior frontal cortex as part of a network that subserves syntactic computation. So for example, here's a quote from Angela Federici: the dorsal back projection from the opercular part of Broca's area to posterior superior temporal gyrus subserves top-down processes relevant to the assignment of grammatical relation. And in a similar, though different model by Ina Bornkessel, and we see a very similar network. One of the difference there is that the direction that is deemed most important for syntactic processing is reversed in Born-Kessel's network, but there too the dorsal connection is the one that should subserve syntax according to these models. From lesion-symptom mapping, earlier studies looking at impaired syntactic performance in post-stroke speakers, we see a vast array of areas that are associated with this problem. So we see inferior frontal gyrus, but also superior temporal gyrus, and specifically for complex sentence production. So that's not just comprehension in general, because there one might argue, well, you know, comprehension can be impaired in many, many different ways, right? You can have a comprehension problem due to lexical access issues, or syntactic, or maybe something completely different. But complex sentence comprehension comes a little bit closer to capturing perhaps agrammatic performance, in the sense that what we're looking at here is particular difficulty with hard sentences, right? So sentences that do not follow the canonical word order that we like. And there we see both research that points at posterior, superior parietal actually regions, temporoparietal I should say, and a study by our own group on Icelandic data showing in fact middle temporal and anterior temporal damage correlated with particular problems with non-canonical sentence comprehension. So the interest, as I said, is first of all aphasiological. What we want to know is whether there is a predictive marker of agrammatic performance. If that is the case, then we may know how to treat it sooner and more efficiently. We may also make use of neuromodulatory techniques, right? So transcranial direct current stimulation, for example, or other ways in which to directly influence and modulate how parts of this syntactic network operate, if it is the case that there is a specific part that is damaged in these patients. There's also a neurolinguistic interest, which is we just want to know what the network that underlies syntactic processing looks like, and that is also relevant for the question itself as to whether syntax as such is an autonomous module of the human cognitive apparatus, right? Or whether it's just a set of functionally independent functions. So the current study. Oh yeah, and throughout this talk I will give a shout out once in a while to my collaborators. The first one here is Julius Fredriksson, who is there left bottom, looking very dreamily. So in the current study we included, in the analysis I included 75 stroke survivors. All of them are in the chronic stage of, well, not all of them have aphasia, so I should just say all of them are 6 months post-stroke at least. As you can see in the graphs, not all of them have aphasia. We think it's very important not to only include speakers who have a particular deficit in our analysis, but also to compare them to people who've had a stroke but who have not been diagnosed with aphasia. The age distribution, as you can see in the graph top right, is centered around age 60. And we have a wide spread of aphasia severity as indicated by the WAB AQ, so that's the Western Aphasia Battery AQ. And the graph bottom right is only for the speakers with aphasia in this group. We did the Northwestern assessment of verbs and sentences with this group of patients. And this was done, and here's the second shout out, by Svetlana Malyutina, who was working in my lab. This test consists of a number of subtests where we pay particular attention to the argument structure production task. So this is a task where the patient has to come up with not just the verb in a picture, but also the action, but also has to name the correct arguments in the correct order. And in order to do away with any influence of lexical access that might be problematic here, or some semantic problem, lexical semantic problem, the words are given. So you get a picture and it will tell you this is a man, this is a ball, and the action is kick, and then the person has to say the man kicks the ball. And if you do that correctly, if you put the verb and the arguments in the right order, then you get a score for the ASPT-A measure, and this is one that will come back later on in the talk quite a bit. We added to that an in-house measure, ASPT-G, which means that the production was actually grammatically correct, because according to the guidelines of the NAVS, you can get a perfect score of A if you just say man hit ball or man kick ball. So we wanted to have a separate measure that also captures whether the production is grammatical. Do we have a question, Grigori?
Yeah, so like if for example, like it's a transitive, right? So it's like it's
they're all transitive, by the way. Yeah, yeah. Oh no, actually that's not even true. Go ahead. Sorry. Yeah.
Yes, I mean, like, is it not right if the right prepositions were used?
You get separate scores for that. So it goes a little far to go into it in detail, but there are separate scores for whether you, if in a phrasal construction, you have to use the right correction. Certainly our G measure captures it, right? So for our G measure, it has to be a correct American English sentence. We added another measure, which is actually inherently part of the NAVS, which is it allows you to look at performance on simple verbs, so intransitive verbs, for example, versus more complex verbs. And we calculated a different score between performance on three-place verbs, so something like give, I give the man a book, versus one-place verbs, I'm laughing, for example, right? So that should capture particular trouble with complex verb production. Then we have a sentence priming production task and a sentence comprehension task. Both of them rely on the same structures. I'll go a little bit in more detail in just a second. We have the total scores of those tasks, but again, like I said before, the total score isn't always reflective, isn't necessarily reflective of any problem with syntax, right? You can bomb on those tests for many different reasons. So we also, again, added a different score between canonical and non-canonical production. And that's based on the following sentences. So the sentence types that are in the SPPT and the SCT are actives, subject extracted WH questions, and subject relatives. And you can see the examples on your screen. And then non-canonical versions of those are passives and object extracted WH questions and object relatives. So in a nutshell, by non-canonical, what we mean is a word order that is different from actor, action, object of the action, right? Theme, if you will. So in the sentence priming production task, the experimenter will say, for this sentence, I could say the dog is chasing the cat, and for this sentence, pointing at the other, the picture, you could say, and then the patient has to say, the cat is chasing the dog. And in the comprehension task, they get one version of those sentences, and they have to point to the correct picture out of the two options. So we divided the stimuli set into canonical and non-canonical sentence types, so we can calculate a different score between these types. To all that, we added what I will call a clinical judgment, and here's my third shout out. Alex Basilakos did a lot of work looking at all the videos of the participants in this study who were describing pictures. In particular, the picture that is used on the WAB, the Western Aphasia battery, which is the cookie theft picture. So both Alex and I, without looking at the NAVS scores or without just looking at the number of the patient and not knowing anything about other test scores, we rated these patients on whether we thought, based on a definition gathered from the literature, these patients should be classified in a binary way as being agrammatic, if we believe in such a thing, or not agrammatic. And a third category, unclassifiable. So in cases where verbal output is so diminished that we can't honestly say whether this is due to some grammatical, or whether it seems to be due to some grammatical problem, or just because of lexical access, or very severe apraxia of speech or dysarthria, for example. We did not score the patients, right? So the score that we came up with, and if we disagreed, we basically came to an agreement. I have to be honest, in some cases, the agreement was that I would make the final call. And we end up with a group of patients who have ones, a group of patients who have zeros, and a group of patients who have no scores, right? And if you then look at what we get, we see that most of the people who are categorized by us as suffering from a morphosyntactic deficit are in the group of Broca's aphasia patients. I should say that we also did a second scoring looking at whether there was a phrase structure deficit, so treating this as a separate, a potentially separate disorder. We found that that mostly converged with the morphosyntactic deficit. So I'm ignoring that for now. I'm only going to be focusing on the morphosyntactic deficit as a binary variable, and I'm going to call that our clinical judgment. So one thing we were interested in is whether the variables on the NAVS cluster together. So whether there's a pattern in some patients on performance on different variables that are part of the NAVS. So we did a hierarchical clustering, entering the variables that I've just mentioned that are part of the NAVS scores, including the different scores that we ourselves calculated and our own ASPT-G score, and what you get is a clustering that looks as what you see on the left of your screen. Obviously you can't read what it says, but next to it, it says morphosyntactic deficit or no morphosyntactic deficit, and I've marked here the patients who have a morphosyntactic deficit in red, and in yellow what you see are the patients that were not that we were not able to categorize. The first clustering is actually pretty nice. We can make a binary division into two groups of patients. Most of those that we labeled as having a morphosyntactic deficit seem to fall in that second group, and most of the others certainly fall in the other group. It's not a perfect correlation, but then of course our judgment was only based on looking at a video based on a particularly, broad definition of what is a morphosyntactic deficit. So it's not a priori given who is correct here, right? It may be that we made the correct call and that the NAVS is wrong. It may, I'm inclined to think that the cluster of well, I'm open to the possibility that the cluster of variables on which people behave similarly on the NAVS is actually a better way, because here we let the data speak, right? So this is what our judgments have in common. So for 48 people, we have the same judgment, and for 5 of them, we have different judgments. And for 7 of them, we both classify as having a morphosyntactic deficit. Then, in order to find out which of the variables on the NAVS were able to make this distinction, we used these two groups as inputs to a linear discriminant analysis. So to find out which variables of the NAVS are able to predict group membership, both on our binary measure, the clinical judgment, and also on the NAVS clustering. What you see in the binary measure is that it's particularly the ASPT-G measure, so our in-house measure that is not really a part of the NAVS, but that does reflect grammatical production. And also better performance on canonical sentences in comprehension. So that's kind of nice that one is able to predict, helps to predict group membership for our binary judgment. Then looking at the NAVS hierarchical clustering, which is of course what the which was based on the same data set, obviously. What you see here is that, again, our G measure is highly predictive, but also the A measure, so whether they're able to put the arguments of the verb in the correct order and so use verb argument structure correctly. Then the total score on the priming production task, and also a little bit oddly, to a lesser extent, better performance on canonical sentences in the production on non-canonical sentences, I should say, in the production task. It's a negative predictor. So, as an interim, to some extent then, the NAVS scores are consistent with our clinical judgment. Right? The morphological deficit appears to be predicted particularly by grammatical production on the ASPT, as well as a comprehension bias towards canonical structures, which is what we would expect, and then group membership based on the subscores on the clustering of the NAVS is predicted by the A measure, the G measure, low SPPT, and an odd bias towards non-canonical sentences in production. Beware, though, all of these scores have very high correlations with general aphasia severity. So the WAB-AQ, which to a large extent is dependent on the rating of the fluency of the patient, and has an outsized, almost outsized effect on the overall severity measure on the WAB. So these measures are very correlated with general severity of aphasia, which is of course something that we need to take into account, and we will. Particularly, we will do that a little bit later. First, I want to talk to you about VLSM, and here's a couple of more shout-outs. So we do lots of different imaging, and some of the speakers in the C-STAR series have already mentioned or talked about some of these. In this particular study, we were looking at lesion. So that's based on the T1 scan and the T2 scan, and the lesions are drawn by Leo Bonilla, who's there in the picture, top right, who is a neurologist, so we assume he knows what he's doing. And then we applied voxel-based lesion-symptom mapping to these data with in-house software devised by Chris Rorden, and lately also with a lot of help from Grigory Yurganov. So I'm very grateful to all of them as well. We use only voxel lesions in at least 5 individuals, so as not to be left with low-powered fringe data. And we use permutation thresholding for the correction for multiple comparisons. So you see the lesion overlap there. That's a standard picture. Doesn't look very different between different studies. For the NAVS data, just looking at the scores on the NAVS and the voxel-based lesion symptom mapping, we can see effects for the A measure, for the G measure, for the SPPT, and for the SCT measure. They're not the same, but I do have to say they are, roughly, they do at least partly overlap. You can see that the G measure for the ASPT is a little bit more anterior, and actually even includes inferior frontal cortex, whereas the simple A measure does not. So there does seem to be really a difference in what these two measures capture, also in terms of the lesions that predict low performance on these scores. We see the same for the syntactic priming production task, where associated lesions are generally more anterior, including inferior frontal cortex, and also bigger, just larger. It's a greater effect, you might say. For the comprehension task, the associated lesions associated with low performance seem to be limited to superior temporal gyrus, particularly posteriorly. Then just looking at whether there's a specific lesion that predicts our clustering and the NAVS clustering, I present you with those pictures as well, and what you can see is that the way that Alex and I classified the patients, the lesions that predict our classification, you might say, are shown in red, and they do include, again, anterior areas as well, even though the bulk of the lesion seems to be posterior perilesional. And then in green you see the NAVS cluster, which is in general a little bit more posterior. But there's a high level of overlap as well, and they're not the same. Obviously these are not the same patients, right? There were a few differences in how we scored. So it's not necessarily it's not easy to figure out which of these is more correct than the other. We should keep an open mind. One that I find particularly interesting is that our scoring of the non-canonical versus the canonical sentence types allowed us to do what is called a partial regression in VLSM. So that means it allows us to only look at the variation that is accounted for by one of these variables, taking into account the other variable. And what you are then left with is we see particular problems with non-canonical sentences predicted by the lesions in green, which are posterior superior temporal gyrus, and particularly the white matter underlying those structures. And that is in line with earlier studies, if you remember, particular problems with complex syntactic processing in comprehension. So we see this only in comprehension. On the production task, we don't see a difference between these two measures. So a lesion in the posterior superior temporal gyrus predicts particular difficulty with comprehension of non-canonical structures. We have another tool at our disposal, which is to look at the connectome. We can look at the connectivity between different regions rather than just the structural lesion. So for that, we use the same data set, but now we parcel out the brain in regions of interest according to the JHU atlas, so to limit the number of comparisons we need to do. And then again, we use permutation thresholding with 4,000 permutations. And these data are based on diffusion tensor imaging that was also collected on the same group of patients. So just to give you an example, the connections, so damage to these connections is associated with low performance on the A measure of the argument structure production task. What you see here is, so we're basically looking through the brain here. We see both left hemisphere connections and also a lot of right hemisphere, so interhemispheric connections that are affected by this particular. No, I should say it the other way around, of course. The performance is affected by damage to these particular connections. Here we have the lesion that was associated with low A scores projected onto the same image. And just to give you a clearer view of just the left hemisphere connections, you can look here. So what we see is that, first of all, the take-home message, which most of you listening don't need to take home anymore because it's already stored at home, is that it's not just the structural lesion itself that predicts low performance, but there's the structural lesion itself has a widespread effect on connectivity across the brain, also extending into interhemispheric connections, right? So that's really illustrated by this picture. So we can look at the scores on the other subtests of the NAVS as well. Most of it shows that there's a lot of connections that are affected, so many that it doesn't really make a lot of sense to now list all of those here. That's not really going to be insightful. So in all these cases, the message is the same. A structural lesion has large effect on connectivity throughout the brain, so certainly we need to look at that. Hold on though, because I just pointed out that the behavioral scores were very much correlated with severity of aphasia, right? And we also know that the severity of aphasia, the overall impairment someone has, is highly correlated with the lesion size. And that's not particularly interesting. So basically, the larger your lesion, the greater your brain trouble, the higher the chance that you have a more severe type of aphasia, right? As well as many other problems likely are likely to be more severe with a bigger lesion. We can see the correlation between the WAB-AQ and the lesion size right here in the picture. It's obviously negative. So how do we deal with that in our VLSM, right? We have to take that into account. So what we chose to do is, rather than take the WAB-AQ measures and use those as regressors to our analyses, because those measures are are basically drawn from the same behavioral data set, right? The WAB-AQ is also very much reliant on the fluency score, and the fluency score is of course very similar to what we're doing on the NAVS. So we decided to do it more cleanly and to take the lesion size itself and to regress out the lesion size from the behavioral variables. So we do this offline. It's possible to do it in NISTAD, and I know Grigori and Chris have already talked a lot about different ways of doing that, but I wanted to abstract away from that for now, and I've just regressed the behavioral data. So I'm using new input to my VLSM analysis. I'm using the regressors, so what is left after I covary out lesion size. And if I do that, we are left with very little. We are left primarily with some overlap in a little area, posterior superior temporal gyrus, the underlying white matter, pretty clearly a ventral part of the connection between superior temporal gyrus and more anterior regions, both anterior middle temporal gyrus, but also inferior frontal gyrus. So we're looking at a ventral point of damage. It's very small. So here we get into a discussion about whether your glass is half full or half empty, which is what the fish are doing in the picture. Oh, you can't read that probably, okay? So the one fish says, whoa, half empty, definitely half empty. And then the other fish, guess who? Just listen to you, always the pessimist. So most of the variance then on the NAVS is accounted for by lesion size, which is accounted for by aphasia severity, which is accounted for by lesion size in the correct order. However, we do, and we do see a distributed network. So we don't see a particular one region that accounts for low NAVS performance overall, right? We clearly see differential behavior on these subtests, and that differential behavior is also associated with different types of lesions. So we're looking at a distributed network of different types of syntactic computations. I've shown that for you in the picture where I've overlaid the four main effects of the on the NAV subtests onto the same image, and you can see that it's a highly colorful image, which means that we're not talking about the same regions. However, we do see some non-canonical comprehension problems that are associated with damage to the posterior superior temporal gyrus. And the overall performance on the NAVS after we regress out that annoying lesion size effect, right, is predicted still somewhat by damage to the ventral route, so posterior superior temporal gyrus ventrally. We did a new linear discriminant analysis because, of course, our behavioral measures are also affected by the severity, so we did the linear discriminant analysis just on the behavior after having regressed out the lesion size, and what we are left with then is 83.9% correct prediction, which is not very high, but it's okay, based on our ASPTG measure, so grammatical production. So predictive of our binary judgment, the judgment that Alex and I made. And then for the NAVS hierarchical clustering, we end up again with the G measure. So that one really seems to be the strongest predictor of the difference between speakers that could be binarily labeled as being agrammatic or having a morphosyntactic production deficit versus not having one. The ASPTG measure is really the strongest predictor for those. Along with that, for the NAVS hierarchical clustering is also just a number of paraphasias made on the ASPT, so number of word errors, which wouldn't necessarily or shouldn't necessarily have to do with syntactic production, at least not directly. Okay, so a new shout out, because Christy Canali, who works in Julius Fredrickson's lab, has done a lot of work analyzing picture descriptions and transcribing these productions by the same group of aphasic speakers transcribing them and putting them into the chat format, which is offered through the Aphasia Bank, and the link is there if you want to know more about it. 3 types of pictures were described by these patients. One of them is the cookie theft, which is part of the WAB, the Western Aphasia Battery. Then there's the picnic picture and the circus picture. One of them belongs to the BDAE, which is the Boston Diagnostic Test, and I believe the other one is part of the Apraxia Battery for ABA2. These are the many variables that we took from that data set, right? So we analyzed with a few in-house measures, so measures that aren't usually part of the chat coding system, but we added those. I'm not going to name them all, but you can see that we looked at ratio of production of different word types. We looked at number of verbs per utterance, including and excluding auxiliaries and participles. We looked at ratios of participles in general, present and past. We take the standard measures of mean length of utterance, which is usually also a strong measure of how complex someone's speech can be. Words per minute, which is a direct reflection of fluency. And if you have any questions about other measures there, please ask. That's perfectly fine. We also calculated the noun-verb ratio, which is a different way of looking at the verb ratios and the noun ratios. And particularly, what will be expected from the literature is that speakers with agrammatic performance should have a bias towards use of nouns, so they would have a high noun-verb ratio, for example. We did linear discriminant analysis based on the clustering that we had before, and as well as on our binary judgment with this data set. What you want to do first, however, is to take out variables that are too highly correlated. So we looked at variables that had a correlation higher than 0.8. We took those out just to make the analysis a little bit simpler. So we're ending up with 25 variables that none of those have a correlation with another one that is higher than 0.8. What we see then, based on the picture description data, is that the ratio of utterance errors, so those are in chat usually marked as plus gram, which is a grammatical error, which is very similar to the measure that we have on the NAVS, right, the ASPTG measure, and then the average words per minute, so fluency. That's basically what predicts the judgment that Alex and I made, the clinical judgment into the binary categories. The NAVS hierarchical clustering is a little bit different. We do see words per minute there again, but the other measures mostly have to do with the ratio of particular words. I only noticed this as I put it up. The signs of those variables are the other way around. So because for the NAVS clustering, the second group is the non-impaired group, whereas for the binary clustering, it was the other way around. So you should look at, so that's why the sign for the words per minute is the other way around, right? So fast speech makes you part of the healthy group. That's how you should look at the right-hand picture. High use of auxiliaries, so am, have, be, makes you part of the healthy group. A high type token ratio makes you part of the healthy group, but low verb inflections, low numbers of past participles, right? So high numbers of past participles, walked, make you part of the impaired group. And this is actually something that we notice in these picture descriptions, that people will say man walking, man walked, you know, so we do see some verb inflection there, if you want to call it inflection. Prepositions, low numbers of prepositions make you part of the impaired group. High numbers of adjectives, so more descriptions relative to other kinds of words like blue, red, nice, maybe. and low numbers of pronouns. So the speakers who have a deficit tend to use a lot of pronouns like he, she, rather than giving a description of the actual person, like man or kite, etc. Now, I'm going to spare you for now to do the same thing and look at all of the data that are associated with the picture description variables. I'm going to save that for later, other than saying that the picture is very much the same, right? So if we look at these, at the measures independently without regressing out the lesion size, we do get a lot of effects. If we then use the regressors after doing an analysis with lesion size, then we are left with way less. What you see here is overall the NAVS plus the picture description measures, all of them on the same brain, and what you should take home from that is that the damage that predicts these different problems is really widespread, right? Again, pointing to a distributed syntactic network, not a particular region that predicts a morphosyntactic deficit in a binary way. That's way too simplistic. I do have one last analysis to share with you. What about an agrammatic pattern of performance based on several measures? So is it possible that if we look at all these variables, the NAVS and the picture description, all those measures that we took, and to see whether there are variables that that can be asked to behave similarly? I'm not explaining that correctly. Whether there are patients that show a particular pattern on groups of these variables. And in order to do that, we conducted a principal component analysis. We scaled all the variables so that they all are, they can all be treated equally, you might say. This resulted in 9 components, so 9 different components. It's a way of simplifying the data in a sense, that together accounted for about 76.1% of the variance, which is not a lot, okay? But there we go. The first component is highly correlated with lesion size. It basically, and that's a negative correlation. So it basically captures normal performance. These, the variables that load onto this component particularly highly are the variables that show fluent speech, no errors, good NAVS performance, nothing wrong here. Second component is actually a group of people who are a little bit better, who are speaking in a more complex way. So they have an excessive number of verbs per utterance, or different markers of particularly complex syntactic production, higher MLU, for example, and still good NAVS performance. They don't have a problem. They are just, more fluent, you might say. This one is also still negatively correlated with lesion size. Our third component is the one that we embrace. This seems to be reflective of poor syntactic production. I've listed for you on the screen the variables that load onto this third component with a correlation that is higher than 0.2, a weight that is higher than 0.2 or lower than -0.2, right? So I'm not concerned with all the ones in the middle. And what you see there is noun-verb ratio, ratio of nouns, morphological agreement errors, utterance errors, and the ratio of other morphological errors. Those are all highly predictive here, or those load onto this component, right? And negatively, the ratio of auxiliaries, the ratio of conjunctions, which is another measure, another way in which to make longer sentences, the ratio of adverbs, the ratio of pronouns are all very low on this one. So we're pretty confident that this component is capturing what might be called, asyntactic performance or problems with syntactic processing. Right? So what we're going to do, and another type of support for that is given by the t-test, where we looked at the different loadings of the third component by the individual speakers categorized as either having the morphosyntactic deficit or not having the morphosyntactic deficit. You can see there that the two groups really differ where it comes to the individual participant loadings for this correlations with this third component, and that's not the case for the other components. So we enter the participant loadings on component 3 into VLSM and the connectome analysis, right? Now we're going to find out whether there's a region or a set of regions that predicts this component, and that would be really nice, right? So wait for it. And this is a barren landscape. So there's no particular lesion type, size or site associated with this third component. And I don't think that's just a null effect. I think that is a real effect in the sense that we see from our behavioral data, this has been the pattern throughout this, right? That in our behavioral data, we do see that certain people cluster together in their behavior, which you might call agrammatic, or they have trouble with certain syntactic subtasks, even more trouble with non-canonical sentences. But they do not share a specific neural substrate in their lesions, right? And that's the effect that, or the lack of it, that we see right here. So to conclude, behaviorally, that's basically what I just said. Agrammatic performance can be captured with NAVS and picture description analysis, and that's relevant also for treatment. So there, treatments are being devised as we speak and are already being worked on and already used to particularly target syntactic, comprehension, or production problems. So these data don't mean that that doesn't make sense. Okay, these data don't really bear on that issue. If someone has a particular problem, you can target it with a particular type of treatment. But it must be noted that many low scores on syntactic subtasks, also on the NAVS, but also in picture description, are highly correlated with general overall severity, and that does say something about the status of a syndrome of agrammatism. Something that I haven't mentioned yet, but which I feel I should mention before I close, is that I was reminded when we were doing this analysis, particularly the binary judgment, that the picture description task is really not a good measure for capturing syntactic production. So many speakers where you just know they can do better, they take an approach where they just name items in the picture description, and even if the experimenter or the therapist says please speak in full sentences, people don't really have to complete the task of picture description. Whereas if you have to tell a story, or if you're spontaneously speaking to someone, that might be different. So currently what we have going on in the Aphasia lab, again with Julius Friedrichsen, is that we're also analyzing the Cinderella stories that we have collected from these same people, and then we're going to see how that holds up. Neurologically, there's no evidence for specific damage to underlie agrammatism as a cluster of behaviors in a group analysis. Okay, this is not a particularly new or revolutionary conclusion, but it does confirm what most researchers, I would say, in this field are currently saying. Right? So it's not the case that we're dealing with a homogeneous syndrome. We are dealing with a set of behaviors where performance can be highly variable between participants and even within participants. That's been shown. However, damage to posterior superior temporal gyrus and underlying white matter, in particular the ventral route, is a point of interest for those of us interested also in the syntactic network, but even perhaps for aphasiological reasons. As this kind of damage appears to predict low performance on task with a syntactic component. And we see that particularly in the ASPT-A measure, and after we regress out lesion size, and also in the particular problems with non-canonical sentence structures in comprehension. Now this is interesting because the ventral route in some of the models that I discussed way at the beginning of my talk, which now seems hours ago, would say that this ventral route is particularly subserves semantic processing in a way. So either that sheds new light on what exactly is involved in these particular tasks, or it sheds new light on these models. So I think we should go from there. And I want to end right there. Any questions from the room?
So, fantastic work, Dirk. I really enjoyed this. You and I have talked about this a lot, and now some of these things I haven't seen before. You obviously did this like at 3 o'clock this morning, maybe. But it seems to me that this posterior cluster, if you will, that you're identifying with the NAVS, when you factor out the lesion sign, there seems to be something there.
Right. It's consistent. Yeah.
Right. So I wonder if you were able to maybe get a lesion location that is common, and there are different ways that you could do that. I wonder if just damage to that region by itself, because it would weigh up your power, whether that wouldn't predict your third component that you get in the PCA.
So, like a region of interest.
Yes, only with one region. Because it seems to me that you do the whole brain analysis on the third component. Implicitly, you've already factored out the lesion size in the first one and some in the second. But this third one, I mean, I would love to say that it's not a coincidence. Therefore, does that make sense?
It does. I'm trying to think if you would do that and you find something, you do want to show that that doesn't go for other areas if you look at that as a region.
So you could have a control
region. Because there are these general effects,
yeah. Yeah, so you could have a control region.
Right.
Just,
yeah.
I don't know, something reasonable.
Yeah. A few control regions.
Yeah.
Yes, oh, sorry. The question was that this region that is identified posteriorly underlying white matter, whether it would be possible to look at this as a region of interest and then see to what extent it predicts the third component that I just showed. So, and we agreed that that would be really cool, and then but that we would need some control regions as well, because we still have that general problem that any damage just basically means more trouble. Right?
But because it doesn't correlate with lesion size, but you would still want to do it, obviously, to have the control.
Right.
Yes.
But yeah, no, it certainly is strong candidate. Yeah. Gregory.
And this basically is to follow up on Julius's question, right? I'm just thinking of other ways to visualize it, right? So, so basically this posterior, temporal junction-ish region, right? So basically damage to this region predicts agrammatism irrespective of how much of the rest of the brain is damaged, right?
It's more anterior to that. I wouldn't call it parietal.
Okay.
But yes.
Yeah. So, okay. So first, if we can actually find the actual ROI, like the, brain region that kind of like fits that bunch of voxels that we find, right? And I don't know which atlas we could use for that, but we can look at all of them, right? and then basically I'm thinking about maybe like a two-dimensional plot where you have, like lesion size versus damage to this region. And if you plot people who have and not have this agrammatism in different columns or something, see where they fall in the plot. It's going to be a very hard question to repeat. Yeah.
I'm
It's a comment.
Well, so that's very similar to what Julius was just suggesting, right? So, but instead of looking at the principal component, to then basically again use this area as a region of interest and now do these analyses anew. Yeah. No, I think that's that is certainly very. I mean, it's feasible. I don't think that's a problem. So what you're suggesting is to look at whether they predict membership of the morphosyntactic deficit or not. Right? As a region of interest. Again, I think the answer is the same. That it would be really important to show that this does not work for individual regions elsewhere if you treat them as a region of interest, because there is this power issue. A lot of this may be under the surface, right? And if you treat something as a region of interest, you suddenly have way fewer multiple comparisons. So that increases your power. But obviously, I want to do do it.
Yeah.
But, yeah, I think that's feasible. Yeah. Who's that?
So quick question. This is probably a more question for Gregory. In terms of like when you regress on the volume size before you feed in the data to the neostat, would you expect to see different results compared to when you feed into neostat the raw data and let the neostat regress on the volume? I guess my question is like, are these two different approaches mathematically, computationally different? Would you expect to see differences in the regions that survive the test?
So just to repeat the question Roosevelt asks is, I regressed out the lesion size from the behavior basically offline, outside of NESTAT, outside of our VLSM analysis. There's a possibility in NESTAT to do the same thing. Are these mathematically the same? And that's a question that Grigori can answer much better than I.
I would think that. Yeah.
Yeah?
Yeah.
So neostat is doing pretty much like the same thing. It just like works on the raw data or regressing out the volume size.
It does it first on the behavior and then puts it in.
Okay, so it's pretty much like the same.
It should be the same. I didn't try it this time, but
Yeah. Friedman lane could be slightly different if you use the, you know, like the Friedman lane option, but like most of it, you know.
so for those of you online, the discussion is now the Friedman lane method, which was the method used for partial regression, may yield some different results if we regress out the lesion size in NESTAT, but we honestly don't know.
Probably not.
Not at this moment. Julius?
So one quick question. So if you don't mind scrolling back to the results from the VLSM with your and Alex's clinical judgment.
Without the regressive, right?
Yes. So where it overlaps with the NAVS results.
Here?
Is that it? Yes. So there's overlap there, certainly. Did
Yes.
you run this as a multiple regression? Just
No.
to see,
No, these are
because
separate.
that anterior cluster
Yeah.
seems very independent to me.
Yes, so I think that's certainly possible, and I think you're absolutely right that what you would get if you ran this as a partial regression. So the question is, did I run it as a partial regression? That was, and the answer is no. If you do that, I think it's highly likely that you will get an anterior region that loads more onto, that is more predictive of our classification, right? And a more posterior region, what you see in green there, that is more predictive of the NAVS clustering. And of course, by definition, if you use a partial regression analysis, they can overlap, right? So that would be an artificial way of separating them out. But yeah, you can. Basically focusing on how different they are rather than what they have in common, which is what I do with the yellow and the rigidity set. Do we have questions online?